Hello, welcome to the Garvan Institute's annual Breast Cancer Public Lecture for 2025. It's wonderful to see quite a full auditorium. Must be at least 150 people we've got here, and I believe we've got many more than that online over Facebook and Zoom and so on. So welcome at home, welcome in the auditorium. We've got a really exciting afternoon of speeches and information for you today. My name's Alex Swarbrick. I'm a breast cancer researcher here at the Garvan Institute, and I'll be hosting this meeting for you. But before we start, can I just ask everyone to put their phones on silent? Thank you. So in the spirit of reconciliation, I'd like to acknowledge the Indigenous people who are the traditional owners of this land, the Gadigal people of the Eora Nation, and we pay our respects to their elders past and present, and any Aboriginal or Torres Strait Islander people here today. So we're here to talk about breast cancer medicine and research. And the reason is that today in Australia, more than 40 women will be diagnosed with breast cancer. Now, 30 years ago, at least a quarter of them might expect to die of that disease within the first few years of their diagnosis. Today, that number is down to less than 10%, particularly here in Australia, where we do very well in the management of breast cancer. And that progress has been made primarily through improvements in screening, so our ability to detect the disease, but also in our ability to treat it. Examples being Herceptin and tamoxifen, really remarkable drugs in the management and treatment of breast cancer. But nonetheless, we are only partway down our journey in conquering breast cancer, and still, unfortunately and sadly, we might expect on a typical day in Australia for 9 women to die of breast cancer, which is a really shocking number. And of course, that doesn't count the kind of other impacts of the disease on patients, their families, their loved ones, and so on. And you know, perhaps unlike some other cancer types, breast cancers are often diagnosed in young women in their 30s, 40s, 50s. These women are in the prime of their lives, highly connected to their families, their workplaces, and so on. So the impacts and ripples of breast cancer are felt hugely by our communities and individually. So for that reason, the Garvan Institute of Medical Research has one of Australia's largest breast cancer research programs in Australia, and our scientists look at everything from the very beginnings of breast cancer, how it forms and how it develops, through to why and when it might spread to other organs in some women, and then finally how we can, after diagnosis, how we can improve our ability to treat those cancers to give women a healthy, happy life after their diagnosis of breast cancer. So today is our annual outreach to the community, to all of you who support us and that we interact with. And you know, for us, it's a very special opportunity to talk to you about what we're doing and to bring you inside and to share with you our kind of hope for discovery and improvements in medicine. I'm also really pleased to announce today that we'll be awarding a new research grant to a young investigator who's undertaking really amazing, innovative research to advance, we think is going to make a real impact on breast cancer research and treatment. This winner was selected through a competitive peer review process, so a really prestigious award, and you'll be the first ones to know about who's receiving that today. So that's really wonderful. That award is really generously supported by the Estee Lauder companies who've been supporting us through this meeting since its very inception. It's wonderful to have many staff members from Estee Lauder companies here today, faces I've come to know over many years, and their support has been really unwavering and wonderful. So with that, I'd like to now invite Hayley D'Onofrio, who's the Corporate Communications Manager at Estee Lauder Companies, to please come up and tell us a little bit more about Estee Lauder Companies' breast cancer campaign.
Thank you so much, Alex, for the warm welcome. So as mentioned, my name is Hayley D'Onofrio and I'm the Corporate Communications Manager for the Estee Lauder Companies in Australia and New Zealand. It is my great honour to be here on behalf of the company as proud sponsors of today's annual Public Breast Cancer Lecture and to present the Estee Lauder Companies Breast Cancer Research Award. 2025 marks an incredible 33 years of the Estee Lauder Companies Breast Cancer campaign. For those of you who may be unfamiliar with how the campaign came to be, it was established in 1992 by the late Mrs. Evelyn H. Lauder, our founder, Estee Lauder's daughter-in-law. Driven by her vision for a breast cancer-free world for all, Mrs. Lauder was renowned for her commitment to advancing women's health, addressing inequalities, and her relentless pursuit for a cure for breast cancer. Her enduring dedication and that of the Lauder family continues to underpin the campaign's ongoing success. Today, the Breast Cancer Campaign is the Estee Lauder Company's largest corporate social impact program and has raised over $144 million globally to fund life-saving breast cancer research, education, and medical services. However, as Alex mentioned, despite the progress over the last 3+ decades, we know that there is still so much work to be done. And it's for this reason we are deeply proud of our enduring collaboration with the Garvan Institute, a leader in innovative research and the advancement of new breast cancer treatments. Throughout this year, and in particularly during October, being Breast Cancer Awareness Month, the Estee Lauder Company's employees around Australia have come together to fundraise for the campaign. I would like to acknowledge our Breast Cancer Campaign Committee, some of whom have joined us in person today and also attending virtually, for their dedication and commitment to this year's fundraising. It is through their efforts that we are able to provide the Estee Lauder Company's Breast Cancer Research Award. This award and monetary grant supports early-stage breast cancer research, allowing those starting out in their research careers to undertake testing a novel, early-stage idea with applicability to breast cancer. This funding enables these researchers to gather preliminary scientific and research evidence needed to apply for competitive, peer-reviewed funding and government grants. This year, we are honoured to award this research grant to Dr. Hani Kim, whose research project is investigating the molecular switches that trigger a PUCBEC3 activation in luminal breast cancer tumours. The hope of her work is that understanding this early process could reveal regulatory events that distinguish these tumours from others, offering a unique opportunity to develop biomarkers for early detection or strategies for the prevention of breast cancer. On behalf of everybody at the Estee Lauder Companies, we would like to extend our thanks to the team at Garvan and offer our sincere congratulations again to Dr. Kim. We hope that this research grant enables promising outcomes, and we look forward to seeing the progress of your work. Thank you.
Thank you, Hayley. Thank you so much, Estee Lauder Companies. Really fantastic support. This is the kind of support that young scientists need to get a great new idea off the ground and to really snowball into something substantial. So it's so important having your support. Thank you. So I'm really pleased now to introduce for you someone who's going to tell us about their own lived journey of their breast cancer diagnosis. Her name is Luann Lawrenson Woods, and she's very kindly offered to come and spend some time with us and tell us about her journey from her diagnosis through her treatment and where she sees her future. So please, Luann.
Hi. Thanks to the Garvan Institute and Foundation, Kate, Alex, Elgene and Liz for inviting me to talk today. When I started to think about what today's update meant to me, I thought about how it's not only what happens in the lab and research that saves lives, it's also what happens in our wider breast cancer community, and how together science and stories can save lives, because I'm proof of that. Because Connie Johnson and her brother Sam publicly shared their story that I found out that I had early breast cancer. So it's especially poignant to be talking here today where the story of Connie and Sam and the Love Your Sister Village come together with science in the Connie Johnson Breast Cancer Research Lab. After Connie died at the end of 2017, Sam went on Channel 10's The Project. I was watching, and while I can't remember what he said, I learned that mammograms were free for women over 40 with breast screen. I'd been generally unwell for a year or so, and I didn't notice any lumps or bumps or anything like that, but I kept getting chest infections. I was 46, so I thought, I'll just take myself off for a mammogram. It was like I was taking my car to be serviced. It was just a health check I was going to tick off. I was stunned when I was called back for a biopsy, where I discovered that I had a dimple in my bra line in that underboob area of my right breast, which was hidden in the shadows of when I did my breast checks of the overhead lights in the bathroom. When the terror of waiting for biopsy results turned to the horror of a diagnosis, I was devastated, overwhelmed, and completely bamboozled by this cancer world, and all the new language that came with it, including my diagnosis, which I still struggle to sometimes say, so I might fluff it up now, but let's give it a go. I had hormone positive, HER2 negative, grade 3 invasive pleomorphic lobular breast cancer that was multicentric and multifocal. And I actually edited that down a little bit. You haven't got my full pathology. Similarly, I found it really hard to get my head around who was in my MDT, my multidisciplinary team, which was another new phrase for me. I wasn't sure who did what and when between the breast cancer breast surgeon, the plastic surgeon, the medical oncologist, and the radio-oncologist, just to name a few. I'd moved to Australia in 2013 to do a master's in cultural studies, studying everyday popular culture and how we make meaning in life. And weirdly, that helped me make sense of my diagnosis and my treatment. In the film and TV world, the word treatment means an outline of a script or a screenplay or a story when it hasn't really been written in full. So I thought of my cancer treatment as a movie franchise. And at first I thought that the specialists were the stars of the show. But then I thought, no, hang on a second. I'm the star of the show, and they're the directors. And throughout 2018, they directed me through chemotherapy, a skin-sparing mastectomy, and radiotherapy. And then in 2019, I had a DIEP flap reconstruction when tissue was taken from my tummy and used to reconstruct my breast. In 2020, I had the second phase of that surgery, which was combined with an oophorectomy and a hysterectomy. And there was a large production team all across Sydney that helped me, allied health professionals who supported me as I recovered from those treatments and that surgery, including here at the Kinghorn. I was mentally undone by my diagnosis in 2017, and at first would catastrophize about all the what-ifs. But as I started to really understand the unknowns that you experience after a breast cancer diagnosis, like waiting for scan results, pathology, surgery margins, or if you're going to get this side effect or you're not going to get this side effect, I realized that I couldn't assume and catastrophize the worst. Inspired by the lyrics of Natasha Bedingfield's song, I decided that nothing was written until it had actually happened. It was all unwritten. And I began to focus on the what is happening now, what is known now, and what is there that I can do to help support my MDT and put the me in the MDT. Like exercise, which my breast surgeon pretty much prescribed for me when I was diagnosed. And I exercised all through treatment and on the days of chemo, when I could, couldn't always, when I could, with an accredited exercise physiologist. And exercise remained a really key part of my prehab and rehab for my treatments and surgeries. But as I shared my story on Instagram under the name Unwritten Me, it became clear that not everybody had the same access to the evidence-based information that I had and could have those discussions with their treating team. And not just about supportive care like exercise, but also their surgery options that were available to them. So in 2021, I launched a series of Instagram live Q&As with surgeons on Instagram where anyone could ask them questions about reconstruction. And in 2022, that evolved into Rewritten Me, Australia's first podcast about reconstruction after a mastectomy, where I talk to leading surgeons and healthcare professionals, as well as the incredible women who've had a mastectomy and share their stories with us. But part of that podcast, I heard from women, many women, that felt that they couldn't ask or curiously challenge the treatments or the options that had been recommended to them as part of their care. They didn't feel confident to speak up. And that was no matter what their background was, their healthcare setting, or how open and wonderful their team was. So earlier this year, I launched Rewritten Me. Sorry, I didn't. I launched Regarding Me. I knew there would always be a problem having them all very similar names. So earlier this year, I launched Regarding Me, a podcast that supports women to speak up and advocate for what's important to them and to be involved in a two-way shared dialogue with their treating team with evidence-based information. A couple of months ago, I was tipped back into the what-if state of mind. I met Sam Johnson, and I finally had the chance to thank him for helping save my life. Because if I hadn't seen him on the telly in 2017 and taken myself off for a mammogram, it would have been 4 years before I would have been called for my first routine screening. And my diagnosis and prognosis could have been very different. But I'm very fortunate that that isn't my story. Still, being asked to speak today has nudged me into another what if. And it's a what if that I work really hard not to think about too much. It's a what if my cancer comes back. And when I think about that, my hope has always been this: that if it does come back, it happens far enough down the track that the work that's done here in these labs and the labs around the world will have caught up, and that they'll have rewritten the treatment for breast cancer. And I know that I'm not alone. The work here is the hope behind all the what-ifs of women who've already had a diagnosis and for those that will, unfortunately, come after us. Women who feel a lump, notice a dimple, feel a puckering, just feel something isn't quite right, or they're called back after their screening. People who fear, what if it is breast cancer? Because through the labs and through research, they have a chance to create a new what if. What if breast cancer was a curable disease? So from me to everybody here at the Garvan, thank you. Thank you doesn't feel big enough, but it's all that I have. And I extend that thank you beyond these labs and beyond the team that work here and outside of this room. I thank all the women who, like Connie, share their stories forward to raise awareness and funds for breast cancer research, especially in October. And I thank all the women who share their experiences, their hopes and their fears in research focus groups, or who share their bodies and their precious time in clinical trials. And thank you to the Garvan for hosting events like this, events that help our community understand the meaning of scientific research, who make it meaningful and more human for us. And to companies like Estee Lauder, who support these moments where we can come together, connect, share science and stories, because today is where our book begins. Thank you.
Good afternoon, everyone. My name's Elgene Lim. I'm a medical oncologist at St. Vincent's, and I'm also the head of the Connie Johnson Breast Cancer Research Lab at the Garvan. We heard about Sam and Connie, but for those of you who do not know this story, Sam and Connie Johnson started the Love Your Sister movement. Luann mentioned how that movement played an integral part in her diagnosis of her breast cancer. Connie passed away in 2017. Connie was awarded the Order of Australia a day before she passed away. And she, like many of you here, was a mother, sister, friend, wife, and she has left an incredible legacy through her philanthropic work, which has led to a legacy here at the Garvan, which we're proud to share through today's lecture, the Connie Johnson Memorial Lecture, as well as the laboratory which I lead here at the Garvan, the Connie Johnson Research Lab. Sam, many of you might know Sam a bit better, starred in Molly and Secret Lives of Us. He was awarded also an Order of Australia. So how's that? Two siblings having an Order of Australia. And he was also awarded a Gold Logie. I can't remember for what, but yes, very accomplished. And he chose to give up his acting career to pursue philanthropic work to raise money for cancer research. So before I introduce to you our winner of awardee of the Connie Johnson Memorial Award for 2025, I'd just like to play a video of Sam.
I tell Garvan's story everywhere and all the time because it matters and because the people need to know about it. I'm very proud to do so. I'm proud to call myself Sam, Connie's brother. I'm proud to be the co-founder of Love Your Sister, but I'm very, very proud of the fact that Love Your Sister's most long-standing relationship with the research agency is with Garvan. That's for a reason. It's nothing but love, always. Thank you.
Shireen, can you make your way up? Professor Shireen Loy will deliver this year's Connie Johnson Memorial Lecture. Shireen is a medical oncologist at the Peter MacCallum Institute in Melbourne. She's also the head of the Translational Breast Cancer Genomics and Therapeutics Lab. She was awarded the Prime Minister's Prize for Science in 2021. She's a friend, dear friend, colleague, and collaborator. She has transformed the international breast cancer research community's understanding of the way immunotherapy works in breast cancer. And I can't think of a better awardee for this year's Connie Johnson Memorial Lecture Award than Shireen. So thank you very much, Shireen, for gracing us today.
Okay, good afternoon, everyone. Thank you, Elgene. Thank you, Alex and Liz. It's a great pleasure to be here speaking to you all today. As Elgene has already introduced, I'm a medical oncologist, so I'm a cancer specialist. I treat only breast cancer patients and have done so for the last 20 years. I'm also a cancer researcher. I run a lab, and we've been really focused on trying to understand how the immune system interacts with breast cancer, essentially, how we can manipulate the immune system to fight against breast cancer. And I'm going to share with you today some of the research we've been doing to try and understand how the immune system also influences the development of breast cancer. And really importantly for women, how life events for women can really reshape our immune system and our general health. So this is what I'll speak to about today. So I think, we really understand why we're here. Breast cancer is the most common cancer in women, and unfortunately the biggest killer of women between the ages of 35 and 65 in Australia and in many countries in the world. What is the biggest killer of men in that age group? Accidental death. So just to put that all in perspective for you. So as we've seen, and this this getting a little bit worse every 5 or 10 years or so. At present, it's 1 in 7 women that will get breast cancer. So if we're sitting in this room, you can imagine what that's going to look like. But more concerningly is that in Australia, actually, our cancer incidence is rising in young people. And you can see there, this also includes breast cancer, but also other cancer types. But women bear this burden because breast is the most common cancer type in women and young women in particular. So breast cancer is a huge burden for society. Okay, how does breast cancer develop? So a long, long time ago, it was noticed that breast cancer had the highest incidence in nuns, actually. And this was then spurned studies many, many years ago, centuries ago, to try and understand and try to work out if nuns indeed had more breast cancer than women who had children. And actually that was found to be the case. And that began research to try and understand whether it was related to hormones and the whole process of pregnancy and breastfeeding that might contribute to a lower risk of breast cancer. This may be relevant to what we are seeing now, because now also we're seeing lower ages of menarche, so when girls are getting their periods, and also women are getting taller as well, and also women are delaying their childbearing till later, as I'm sure many of people would understand. But there's also other factors potentially that could be contributing to this risk, and you've all heard them: lifestyle factors, weight, diet, alcohol, working and sleep, for example, may all contribute to this increased incidence. But what's been most consistent is this relationship with children and breastfeeding. So do people here know about that fact that the more children you have, the lower risk? Yes, and the longer breastfeeding, no? Okay, so this was published a very long time ago, actually, in The Lancet, in a very important medical journal, which if you look at your risk of breast cancer, and it's going down, it's falling, and that's correlated with the number of children that you have. And you can see there, the more children you have, the lower your risk. But in that very large study, they also worked out that actually if you had breastfed, your risk was even lower than the additional risk of the extra child. So breastfeeding and children led to a reduced risk. And what was particularly intriguing to us is that breastfeeding was particularly important for one of the most aggressive breast cancer types, which we know as triple-negative breast cancer. And the reasons for that are actually unknown. So people have thought about this for a long time, and as you can imagine, the main focus has been about hormones. So pregnancy, breastfeeding, etc., changes hormones in your body. You have estrogen, progesterone, prolactin, all of these hormones change in your body as you undergo pregnancy, birth, and breastfeeding, and then ultimately, as everyone who's had children here knows, breastfeeding then becomes involution and the breast returns back to your pre-pregnancy state. So for a long time, people thought this was related to hormones and how hormones change, and potentially how estrogen and progesterone changes your signals to the breast. So I'm sure all the women here know during your menstrual cycle, your breast also changes, right? So you're having this constant cycling of your breast throughout every month, essentially, and this is exaggerated through pregnancy and breastfeeding. So it's long been believed that this has been the main influence with regards to this decreased risk. The other main hypothesis is thought that as during pregnancy, as your breast grows to accommodate breastfeeding, these grown-up cells are generally hard to become cancerous. Another very popular thought, and there's probably some truth to this, is, as you can imagine, breastfeeding and weaning, the breast enlarges as it's required to breastfeed, and then as you end your breastfeeding, the breast returns to normal size. So all that involves cells being cleared out. So, we call this involution, but essentially you are losing massive amounts of cells from the breast, and then the breast regrows. So potentially some of the cells that you are losing may have become cancerous in the future if those cells were remained. So it's basically like a cleaning out of all these damaged cells. And then the other main theory is that as you get older and post-pregnancy and breastfeeding, that your breast doesn't need to grow so much anymore. So these growth signals, that involve your breast changing through the menstrual cycle and through pregnancy are actually diminished. So there's fewer growth signals. So that may be that if you have any cancerous cells hanging around, they're less inclined to grow after that whole process. And so for many years, the explanations have really been about hormones and tissue changes, etc. And also there was all of the evidence about hormone replacement therapy, etc. So we've really been very focused around hormones. But I study the immune system, and it's just been very hard to imagine that your immune system does not have a part to play in this whole complex situation of breast cancer. So everyone knows your immune system, it's the guardian, it's how we've stayed alive over evolution, and it protects us. And there's many parts of your immune system. There's your skin, it protects us from things entering our body. There's what we call the innate immune system. So these are very first responders type of immune system. They're like cells and chemicals that stop invading bugs or pathogens. And then finally, the third level, which is the most sophisticated level, we call the adaptive immune system. And in particular, this comprises mainly of T cells, our little T cell there, our CD8 T cells, and we call them like the soldiers. So they really do the main bulk of the attacking and the getting rid of all these pathogens. And then the nice thing about these T cells is that they remember. So once they've seen something before, they tend to remember it for the future. So, my lab has always believed, and, the cancer has moved towards an era of immunotherapy because we believe that if you can recondition your immune system, potentially they do the bulk of the work, and also they can protect against future recurrence because they develop memory. It's imprinted in your body, and it stays in your body if you can re-educate your immune system to protect yourself against cancer. So we've spent a lot of time studying how our immune system can fight breast cancer. Some breast cancers have lots of immune cells in them, and we've developed a method to look at this using a microscope. And you can see this as a microscope picture of a breast cancer, and those little purple dots, they're our T cells. And in this particular breast cancer, there are lots of them. So some people have a lot of T cells in their breast cancer. In contrast, some women have hardly any. So you can see there's nothing there. And why we know this is important is because we've looked at how these breast cancers do and how the patients survive with and without cancer recurrence. And you can see, and this has been reproduced now in tens of thousands of patients, that if you have lots of immune cells in your breast cancer, you're doing really, really well. And if you do not have any immune cells in your breast cancer, you don't do as well. So the question has always been for our lab, how can we change or boost your immune system in patients who don't have these immune cells and make your immune system activated so you can do better and fight your breast cancer better. Just briefly going to talk about these biomarkers because we've developed this out now over many years. It's really simple. You can look down a microscope, and it's really been implemented internationally in many guidelines. And the nice thing about our biomarker, because it's so simple, you can do it all around the world. So this is a workshop we conducted in India actually last year. These pathologists get paid $5 a day, and yet they paid for my colleague to go over and teach them how to look for these immune cells using their microscope. And my colleague Roberto there, right in the middle there, he said he's like the only white guy that they've ever seen in that little city, but they were really keen to learn this particular method, and I think because it only involves a light microscope, which everyone can access, people are really keen to do it. So you can see there that we have a website and over 87,000 users of this biomarker, because this gives us information about how the immune system or how the patient's immune system is reacting against their breast cancer. Really easy, really simple. And why is that important? Because we can treat it now. So we've learned that if you have an immune response, we can activate it with drugs. And this is just recently came on to the PBS in Australia, But at Peter Mac, we've been using it now for nearly 10 years in trials. This is really emphasises the importance of clinical trials and clinical research because you get access to these drugs 10 years before they're approved by the healthcare system. So we have been involved because we saw these immune cells in breast cancer. We knew that they would be important, and we've worked with companies and also other researchers around the world to develop immunotherapies against breast cancer. And specifically, it works well in triple-negative breast cancer. And now the studies have been completed, and this is why it's approved by Medicare and on the PBS, that this drug, in addition to chemotherapy, reduces deaths from breast cancer by a third. So this is really a big breakthrough for specifically triple-negative breast cancer, and it's really proof of principle in breast cancer that we can activate the immune system to fight against breast cancer and prevent recurrence and death from breast cancer. And so this has been a big breakthrough for triple-negative breast cancer and breast cancer patients in general, and this was approved in Australia in 2023 and used for all patients can get access to this drug. Okay, but the interesting thing along this journey is that actually we found these immune cells in normal cancer-unaffected breast. So what are they doing in normal breast? There's no infection there. And so we asked the question, can they help us protect against breast cancer? And this was just a study that we did. We had primary breast cancers, they were all those purple dots. But normal breast without any cancer, no history of cancer, also had lots of these T cells. So they were there already. So, they're already there. And so the question we asked is, why are they there? And if you recall that relationship between breastfeeding and triple-negative breast cancer and pregnancy, and what happens when women breastfeed? There is mastitis, there is, infection, there is interaction in the saliva, from the baby's mouth. So is that conditioning the immune system in the breast? And the question is, can that protect against breast cancer growth and formation? So we asked the question first, are there actually more immune cells in cancer-unaffected breast of women who've had children compared to women who've not had children? And actually we did find that. We studied over 260 women who'd had breast reductions or prophylactic mastectomies. So this was healthy breast tissue. We studied many different populations, so we believe this data is applicable to lots of different diverse populations, as well as women of average risk, as well as high risk of getting breast cancer. And we found that there were these immune cells in the breast, and there were lots more in women who'd had children previously compared to women who had not. And the interesting thing that some of these T cells were very long-lived. We saw them in specimens that had been collected sometimes 50 years after the birth of their last child. So, these immune cells are really long-lived in the breast. So that was one part of the evidence that these T cells or these immune cells are there in normal breast. But can this help us protect against breast cancer? So to do this, we had to use a mouse model because it's hard to do this in humans. But we did one experiment where we compared a mouse that had undergone pregnancy, breastfeeding, and involution compared to a mouse that had not, and we implanted breast cancer cells. And what we found is in the mice that had pregnancy and breastfed and undergone lactation and involution, their tumours were smaller, or there was not much there at all compared to the mice that had not undergone that. The most important thing is when we took away the T cells or the immune cells from that Paris mouse, this changed. So basically after that, there was no difference in tumour growth. So this was a message to us that the T cells are really the most important part of this whole experiment. And the tumours from the Paris mice were smaller, and they had lots more T cells. And we also worked out that actually the whole immune system of that Paris mouse is different from the virgin mouse. They had T cells that had gone from the breast out to the circulation as well. So not only was the immune system changed in the breast, but it was also changed throughout the body. So the process of having the child breastfeeding, as well as the whole involution process, changes the whole immune system of that Paris mouse. And then finally, we wanted to understand the contribution of breastfeeding. So we did studies in over 1,000 patients diagnosed with breast cancer after their last child and looked to see if they had breastfed or not, and compared those who had children and not. And what we found was that actually in women who'd had children and had breastfed, but then developed a breast cancer, their tumours were smaller and they had more immune cells in them. And we also worked out they did better from breast cancer as well. So women had better outcomes if they had breastfed and their tumours had more immune cells in them. So that suggests to us that breastfeeding adds an extra component of immune protection. So why might this exist? Well, I mean, let's face it, women are essential for the survival of the human race, right? So, you know, they have the children, they breastfeed. Breastfeeding is critical for survival of the human race, and it has to be done in an effective manner. And the survival of the mother is also critical for survival of the human race. So evolutionarily, this makes sense in that the immune system has evolved to protect the mother post-birth and to assure the survival of the baby and the family. But we believe that immune imprinting of that whole process creates memory that also reduces breast cancer risk and possibly other disease types as well and other cancers. You know, women do live longer than men, and that's actually the reasons for that are quite poorly understood. So why does this change how we think? Well, basically now we've gone from a treatment, you know, I've talked about Keytruda and Pembro, and now we can talk about how can we reduce the risk of breast cancer in our modern world, because now we talk about prevention, and now also it changes the way we think about the whole problem, because it's not just about hormones, it's about immunity. And actually, if you think about it, the only other example is like HPV vaccination. You know, immunity can now prevent cancer and not just treat it. So looking ahead, what we're trying to understand is, of course, you know, how does parity actually protect against the breast cancer? Obviously, this has societal implications in motivating breastfeeding to improve not only breast health, but health of the mother. But importantly, this allows us potentially to understand how we can recreate this protection in women who are having children later or deciding not to have children as well. How can we mimic this protection? So the key take-home messages: pregnancy and breastfeeding leave behind T cells in the breast. These T cells are important, and they are also around the body, and this improves our defense against breast cancer, specifically triple-negative breast cancer, potentially other cancers, and also other diseases such as cardiovascular disease and diabetes. And our work, we believe, will result in a shift in how we understand immunity and its role and its involvement in how we can have protection from breast cancer in the future. And I will leave it at that.
I'm sure you'll agree with me, that was an absolutely fascinating, stunning presentation. For someone to be able to do the kind of leading edge research that Shireen does, but then communicate it in a way that is so crystal clear is truly an unusual gift. And we're so lucky to have you here. Thank you, Shireen. So it's my pleasure now to introduce Associate Professor Liz Calden. She's a faculty member and leads a lab here at the Garvan Institute, and she is going to now take us through an update of the last 12 months of research outcomes here at Garvan. Over to you, Liz.
So I've got to say that's a really hard act to follow, Shireen. That was just an amazing presentation, but I will do my best. All right, I'll just... Here we go. So we've all been told already, we know breast cancer is such a worldwide problem. But unfortunately also this year in Australia, it was announced that Australia and New Zealand were actually the place in the world that's got the highest rate of breast cancer. And this is what this looks like in Australia. We have about 21,000 diagnoses in Australia, more than 3,000 deaths, and we have a large number of people who are living with metastatic disease. And also, and something that I know so many of you are familiar with, we have actually so many thousands of people who are living post-breast cancer and in fear of recurrence and living with those symptoms associated with breast cancer treatment. So altogether, as researchers, we've got a huge challenge to really address many of these problems and many of these issues to make life a lot better for everyone in our community, because as we know, we are all connected to people with breast cancer, and that's why I include this image here, because breast cancer does not just affect those individuals, it also affects their families and communities. So today I'm really excited to present on behalf of my colleagues and present updates from across the program of breast cancer research at the Garvan. And I'm just going to focus on 4 things today. I'm going to give an update on our Estee Lauder Award winner from 2024, tell you about a new tool that's been developed and published from the Garvan this year on how to distinguish different parts of a breast cancer. I'm going to tell you about why we think drugs can stop working in estrogen receptor positive breast cancer. And finally, I'm going to share with you an exciting new program which got funded this year at the Garvan, where we hope to see some real inroads into addressing recurrence of breast cancer. So to start with, I'll just talk about the work of Dr. Nicole Uono, who was our winner of the Estee Lauder Award in 2024. So Nicole's interest and project was really exciting. It was to look at this protein called SPARC and how it was involved in chemotherapy response in lung metastasis. So as you know, when a cancer spreads, it's really important to treat it effectively. And there's different proteins we believe may be important in modulating that response. And Nicole's team really thought that this protein SPARC might be important. But cancers are very complex, so they had to find out where that protein was and what it was doing. So as part of Nicole's studies, she was really looking at where that protein was and what it was doing in the cancer. Now the cartoon I'm showing you here is a cartoon of a cancer. And as you can see, it is actually very complex. It's not just made of cancer cells, it's actually made of a whole bunch of cells. When a cancer starts growing, it can do this really incredible thing where it can recruit cells from the tissue around it and start incorporating it into that cancer. Some of those cells are called fibroblasts. And Nicole wanted to know, well, this protein SPARC, is it acting from the cancer cells themselves, or is it acting from the local environment and what it recruits into that cancer? And I'm just giving you a very brief update here, but what Nicole has found is that this particular protein is actually acting from both. And she was able to see this protein, which is the brown stain in these images, is present in both those cancer cells and in the non-cancer cells. So now her team is working to understand what exactly that protein is doing in those 2 environments, and they're preparing a manuscript on these findings. I'm now going to turn to one of the really exciting projects that got published out of the Garvan this year, and this also addresses this issue of just how complex a cancer can be. So this is really looking at whether within a cancer all the cells are the same. So going back to this cartoon here, looking at the cells there that are coloured pink, we think of those as the cancer cells in this cartoon. But we know that even the cancer cells within a cancer are not all the same. And Associate Professor Christine Schaffer, with a lead team member Shabani Gupta, as well as Professor Sarah Kummerfeld, have published a paper this year where they've come up with a really exciting computational tool to help us understand what are the differences in different parts of a cancer. So Shireen's told us about what do we see down the microscope, or what a pathologist sees down the microscope. And this is an image of a cancer, and as you can see, we can see certain elements within it, but it's fairly homogeneous. Like, it kind of looks the same all the way through. But as researchers and oncologists as well know, that a cancer is not the same all the way through. And it's the differences within different parts of a cancer which actually give its ability to not respond to drugs, to spread to other parts of the body. And it's really important for us to dive into that and understand what different parts of the cancer are made of. So what Christine's team did was they looked at what was kind of most extreme in that cancer. They used this technique where they look individually to get qualities of all the cells in the cancer, and they found those cancer cells within the cancer that had the most extreme qualities. And they then developed a computational tool where they were able to kind of spread those cells apart on a gradient and then map every other cell in the cancer along that gradient. to try to figure out how all the cells relate to one another. And this gave us a lot better picture of the complexity of what's in a cancer. So now in this cartoon, you can see that we can start mapping different cells with different qualities all the way through the cancer. Now, this was an actual sample they used in their study, this particular tumour. So I'm going to show you what the data actually looks like from their study. And what they found was that they were able to identify 5 different types of areas in the cancer. And when you overlay these, you can see how that they interact with each other, but they form very distinct zones. And this is a really novel computational tool to be able to do this. But I guess what you all really want to know is, well, what benefit does that bring? Well, it's actually incredibly important, because when we start mapping different parts of a cancer and we know that they can do different things, we can start asking different questions. We can start asking questions like, which parts of those cancers will respond differently to drugs? And which parts of those cancers will actually be responsible for the spread of the cancer later on? And by understanding which are the really serious parts of those cancers, we can start thinking about tailoring therapies so that we're actually able to treat cancers better. So I'm really looking forward to where they take this work in the future, but this is really foundational work that's going to help us understand breast cancer better. The next piece of work, I'm really proud to be able to tell a story from my own research lab. And this is a story about estrogen receptor positive breast cancer, which we study a lot in my group. This is research led by Dr. Sarah Alexandru in my group, where we were trying to understand drug resistance in estrogen receptor positive breast cancer. So estrogen receptor positive breast cancer is an extremely common type of breast cancer. It's about 75% of breast cancers, and it's known as estrogen receptor positive breast cancer because the cells have the estrogen receptor in them. It's a little protein that allows the cells to respond to estrogen, and it occurs on a lot of the cells in the cancer. Why do we want to know this? It's because if estrogen is in the body, the estrogen will go to those estrogen receptors and then allow these cancers to grow. And this is an extremely important discovery. It's something that's been known for decades, and it's actually allowed the development of some of the most important breast cancer therapies that have ever been used, which are the therapies that block the action of estrogen, known as anti-estrogens. And I've just put a box here with some of those therapies. I know many of you in the audience will be familiar with these therapies, but these are all therapies that either directly block the action of the estrogen receptor or block the pathways associated with the estrogen receptor. And they've been extraordinarily successful because they block cancer growth in these types of cancers. While they have been successful, while they've saved so many lives, unfortunately about 30% of people with estrogen receptor positive breast cancer go on to recur. And these therapies stop working, and we get drug resistance in these cancers. And in my group, we've been focusing on what are the mechanisms of drug resistance, what actually changes in a cancer to make it resistant to these drugs. We've been performing DNA testing or DNA genetic screens to try to understand this phenomenon. So every cancer cell's got an awful lot of genes in it, about 25,000 genes. So what we did was an approach where we take out each of those genes and we find out which of these genes are responsible for the cancer cells growing. It's a very intense analytical technique, but at the end we do DNA sequencing and we're able to find the group of genes responsible for drug resistance. And to cut a long story short, what we found was that there was a particular group of genes that I'm going to call the junk alarm system. We found that this group of genes was knocked out in the cells that weren't responding to cancer drugs. To explain what this alarm system is, what this means is that normally when you have a cancer cell, if the cancer drug comes along, this alarm will go off and then the cancer cell will know what to do. It will know it's time for it to die or for this cell to permanently go asleep. However, in those cancers which manage to get rid of this alarm system, we find that those cells are then able to persist in their growth. They keep growing, and that's why we get drug resistance, because these cells are able to ignore the normal death signals of the cell and they keep growing. Again, what do we see as the potential impact from this sort of research? We think this type of research is really important because it gets us to start thinking about how is this going to change impact for patients. So for example, if a patient has their tumour analysed and we find this type of alarm system is missing, and they're being treated with the therapies that are normally used for these cancers, this will make us think about whether those patients need extra monitoring. Do they in fact need more intensive surveillance to see if their cancer is coming back? So that's one of the outcomes of this sort of research. We can also offer alternative therapies straight away when we think that the absence of a gene is actually going to cause drug resistance. And finally, in work that we're continuing in our lab, we're hoping to identify better drugs for patients who are lacking this particular pathway so that they can be treated better in the future. Just in the last couple of minutes, I want to tell you about this exciting new research program we have starting this year at the Garvan Institute. So some of you may have seen this news article. It was in the Sydney Morning Herald in August, announcing the award of a really important new program by the National Breast Cancer Foundation for the All Clear program to address recurrence in breast cancer. So this was an amazing $25 million grant, and this grant has been awarded to Associate Professor Christine Schaffer and Peter Croucher as the lead of this grant, and they're leading a national and international consortium of researchers to address the problem of breast cancer recurrence. They're also integrated with a New South Wales-wide team of hospitals so that they're able to implement clinical studies as data comes out of their program. To give you an idea of what they're trying to achieve with this program, they're really looking at why do certain breast cancers occur and how can we track those cells that are initially undetectable but then go on to grow into breast cancer recurrences. And one of the culprits here is really bone metastasis. So often when people are diagnosed with a breast cancer, the primary breast cancer might be seen, but unfortunately these cells that have escaped from that cancer cannot be seen on scans, and they might be lodged in the bone or in other parts of the body. And at the moment, we just do not know enough about those cells, and we cannot predict if they're going to grow into more cancers, and if they do, when they will do it. So the aim of the program is to address just those issues. They're going to be analysing serial biopsies, bone biopsies, to look at whether those cancers are present, when they recur, and what's the sort of biological pathways that are activated in those cancers. And the ultimate aim from these studies is to really understand what makes those recurrences tick and how can we target them better. And ultimately, the aim of their program is with the title of their program, All Clear. They want to be able to confidently give people the all clear to know that they no longer will have any chance of a recurrence. And that's something I think we can all think is a really amazing goal, and I'm looking forward to the outcome of this program. So I just want to finish up by saying a few thank yous. Thank you to all of you for being here and for your attention today. I think we are not able to do the research we do without the amazing support of the community that we have in Sydney and Australia-wide. So thank you all for being here, but I'd like to particularly thank our consumer advocates who support our research and let us know about what's really important in outcomes for breast cancer, and all the incredible funding bodies which help us undertake the work we do, and especially the Estee Lauder companies for supporting the award today, Tahani, and for our event today, so we're able to communicate the outcomes of our research. Thank you so much.
No introductions needed. I will now give you a bit of an update regarding the clinical aspects of breast oncology. So in the last 12 months or so, we've had a number of new therapies that have been approved by the PBS in Australia. One of them is a drug called trastuzumab deruxtecan, which is a mouthful. It's also known as Enhertu. It was approved for use in patients with metastatic HER2-low breast cancer from the 1st of September last year, and expected to benefit about 2,000 Australian patients per year. And in this particular trial, patients had the hormone receptor positive subtype of breast cancer, but they also then looked at the expression of another protein on breast cancer cells called HER2, and a low expression of this protein was the inclusion of the patients onto this trial. So amongst patients with HER2-low metastatic breast cancer who've received prior endocrine therapy, this particular drug was compared to chemotherapy, and as you can see from the curves, the trastuzumab deruxtecan treated patients did better than those receiving chemotherapy. So this is a new drug that's now available to patients in Australia. Another new therapy approved in the last 12 months or so is a targeted therapy called olaparib. And this particular drug works in patients who have cancers that are unable to repair their DNA. And the patients who do have this issue of DNA repair deficiency are patients who carry the BRCA1 or 2 mutations. So it was approved for patients with early stage, so these are not metastatic patients, but patients with early stage breast cancer who carry the BRCA1 or 2 mutation, and is expected to benefit patients in Australia, benefit about 300 patients in Australia per year. In this particular trial, they compared the drug olaparib with placebo. And as you can see from these curves, the patients who received olaparib had fewer deaths and better survival compared to patients on placebo. Olaparib, the same drug, was also approved for use in patients with metastatic BRCA1 or 2 mutation-associated breast cancer from earlier this year. In this particular trial, instead of comparing it with placebo, they gave this tablet, olaparib, and compared it with chemotherapy. And as you can see from these curves, survival curves, the olaparib treated patients had a better survival compared to patients who received chemotherapy. So this is also now PBS approved. More recently, in fact, as of this month, a new therapy has been approved for use in patients with metastatic hormone receptor positive breast cancer. And this particular drug is called capivasertib, also a mouthful, but also known as Truqap. And in this particular trial, patients with hormone receptor positive metastatic breast cancer who have had a prior line of treatment with endocrine therapy and a CDK4/6 inhibitor, the combination of capivasertib, which is a tablet, with fulvestrant, which is an injection, the patients who received this combination did better than patients who received the placebo tablets and fulvestrant alone, as you can see from these curves. So this is expected to benefit approximately 3,000 Australian patients per year. Another treatment that has been approved for patients with early stage hormone receptor positive breast cancer are these two drugs, abemaciclib and ribociclib. These drugs have been used successfully in patients with metastatic ER-positive breast cancer, and the trials have started to report benefit in patients with high-risk early-stage ER-positive breast cancer. And these two trials have now reported some positive results, and as a result, the PBS has approved its funding in high-risk patients only, and is expected to benefit about 2,500 patients per year. So while all this is great, there are still ongoing challenges in Australia. So one example is that whilst the clinical trial for ribociclib in early-stage breast cancer has shown benefit in patients not only with high-risk early-stage disease, but medium-risk early-stage disease, our funding agency has only agreed to fund those with high-risk disease, and there therefore remains a gap for patients with this medium-risk early-stage ER-positive breast cancer who are unable to access this drug on the PBS. Another ongoing challenge in Australia is molecular testing. So increasingly, we are using next generation genome sequencing to identify targets such as these mutations in the cancer. These treatments can potentially be matched to treatments that target these specific mutations. And as yet, in spite of this evidence, there is still no funded test for Australians. And if you do these tests commercially, they're about $3,500 to $5,000, making it difficult for the majority of Australians. It's currently available through a national program, but this is limited to tumor only. These tests can now be done with a blood draw. That test is not available through this national program. And it also requires a much longer turnaround time, but this is done through a research program led by David Thomas and the company called Omico. So we run a lot of clinical trials here in Australia, and I'd like to acknowledge many of you here in the room and in the audience online that you have been a key partner in this research. We try to run as many clinical trials here as possible, and as Shireen has alluded to earlier, this allows us to access some of these therapies before it even hits PBS approval. Importantly, it helps us advance the field forward, making the world a better place for the next generation of women with breast cancer. So we run a variety of clinical trials. We're not quite the size of Peter MacCallum in Melbourne, but in Sydney we do run more breast trials here than any other site in New South Wales. Now, there are two of these trials that we ran here that have recently been reported, but yet to be PBS reimbursed. So a number of you may have been involved in this particular trial called the Patina trial, where a drug, a CDK4/6 inhibitor called palbociclib, is given to patients with HER2-positive, hormone receptor-positive metastatic breast cancer. This trial had about 500 global participants, 50 Australian participants, and St. Vincent's was the lead Australian site, and we recruited 12 patients onto this study. And this is a practice-changing study. As you can see, the addition of palbociclib to standard therapy actually improves patients' outcomes quite significantly. And this is just with the addition of a drug, a tablet called palbociclib. So this is not yet available in Australia, but we look forward to the drug being approved for this indication here. Another recently completed trial is the WinPro trial, which is the use of progesterone in early-stage breast cancer. So St. Vincent's, once again, was the lead site for this study, a national study of about 200 patients. About a quarter of all Australian patients came from our site. In this particular trial, patients with early-stage breast cancer were randomized to receive 3 treatments: letrozole plus progesterone, or tamoxifen plus progesterone. We compared the marker for cell proliferation called Ki-67 in the pre-treated sample versus the post-treated sample. In our study, the 2 letrozole-containing arms did very well. This is the degree of Ki-67 suppression. Letrozole alone suppressed it to about 88%, and the addition of progesterone increased it slightly. So what are the take-home messages? Progesterone plus letrozole given preoperatively to patients with ER-positive, PR-positive breast cancer led to a similar suppression of Ki-67, the marker of cell proliferation, and the addition of progesterone to letrozole was well tolerated and led to a reduction in hot flushes, and it supports the evidence that progesterone is safe in ER-positive breast cancer, and this was presented by Lucy Hagstrom at an international meeting this year. And every year we talked about this, trying to get this tool called EVA, which we try to get out to the public in order to empower patients and increase their awareness of clinical trials. Kate Soar is leading this, and we hope to be able to launch this using new technology, including generative AI technology, for the translation of complex medical information into patient-friendly language. We've co-designed this with patients and clinicians, and a number of patients I see are in our midst today. So thank you very much for your partnership in this project. And we anticipate to launch this early next year. And finally, I'd also like to acknowledge the many patients in our midst who come to St. Vincent's who participate in Project SHARE, which stands for Specimens Help All Research Efforts. Through your contributions of your tissue, it has allowed us to collect many tissues and generate collaborations internationally to understand mechanisms of treatment resistance, understand mechanisms of metastases, and establish multiple laboratory cancer models in which we can model new therapies on. Thank you all.
Thank you, Liz and Elgene, for those wonderful updates on research and clinical treatments. We've now got about 15 minutes before the end of today's session and an opportunity to ask some questions of the various presenters that we've heard from today. We've had a series of questions come in over Zoom and from Facebook, and so I'll be guiding a bit of a Q&A. So can I please invite all our speakers up to the stage? Thank you. Well, before we get underway, I'd like to first introduce Peter Bryden, a breast care nurse here at the Kinghorn Cancer Centre. Peter, could you, round of applause, please. Could you tell us a little bit about your role, what a day in the life looks like for you?
Hello, everyone. What does a day in life look like for me? Busy. I'm surrounded by all these wonderful patients. I get really emotional. I'm really sorry. I'm really lucky to support and navigate, translate for a wonderful group of women. A few men, but mainly women. I'm really lucky to work collaboratively with some wonderful medical oncologists, some radiation oncologists, and some fantastic surgeons. And I carry a bat phone. I feel like Batman. And that's about it. Sorry, guys. I get really emotional on these days. So, and it's not because of anything other than I'm really lucky to support you. Thank you.
I think many of you would know Peter. Yes, and everybody loves Peter. She wears her heart on her sleeve, and we're very privileged to have Peter providing support to our patients with metastatic breast cancer.
So, Luana, a question for you. You very generously shared your story with us, and your really kind of wonderful perspective you've taken and proactive approach to sharing your experience and kind of drawing a community around you. One of the things that struck me when you spoke was about how important you see it in patients becoming part of the decision-making process. And obviously that can be challenging. We've seen how complex the research can be and the medicine can be. What was that experience like for you, trying to educate yourself and understand the diagnosis that you'd been given?
It was really, really hard. I use that word bamboozling, and that's just what it was like. It was a whole new world, a whole new language. And I think, kind of laughed around my diagnosis when I explained it, but invasive pleomorphic lobular breast cancer. When I Googled that, but there's a reason I Googled. I couldn't find any information in any of the amazing resources that we have here. So, there were resources that helped me, from BCNA to the Cancer Council. I had information, obviously, evidence-based information from my team, but in between that time of getting that appointment with an oncologist, I Googled pleomorphic lobular, and I couldn't find any information about that. And I still find a gap now when it comes to pleomorphic. People are starting to talk more about invasive lobular, but I really don't see anything about pleomorphic. So it was really, really hard. In the work that I do now, I do interview people around, how do we navigate these things? And I spoke to somebody from the Sydney Health Literacy Hub, and they had some great advice for Googling. And it's like, you need to think about, who do you want to hear this information from? And when I read about invasive pleomorphic, I came across a website that gave me the most dire information. And I'd already had people wince at me when I told them what I've been diagnosed with. But then at the bottom of this very, 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 very long website page, there was a statement that said, this information is now out of date. Please refer to something that's more relevant. I don't know why they couldn't have it at the top of that page, but it's really hard to not remember that and the fear that came with that. So it was really, really hard. Look for evidence-based information. There's great resources around there. Do ask yourself, who do you want to hear that information from? Because it's very difficult to unlearn it. But yeah, it was tough.
Thank you. So a question here from online, which is, I think we heard from both Dr. Loy and also Dr. Calden about this somewhat worrying increased trend in diagnoses in Australia. How much of this do we think is breast cancer indeed becoming more common versus changes in the way we detect breast cancer in screening?
Yeah, so we've looked at this, and it's definitely in Australia, actually many women go to screening compared to North America. So there is a degree of reflection of increased uptake of screening in those numbers that we see. So, but we have very low mortality actually relative to the number of cases that are diagnosed. So I don't personally believe it's an Australia phenomenon for breast cancer, at least.
Thanks, Shireen. So while you've got the microphone, you told us about some stunning new research that really turns on its head our thinking about prevention in the earliest stages of development of breast cancer. And, this idea that potentially by improving our immune system, we can reduce our likelihood of later developing breast cancer. So, looking into the future with your crystal ball, how big an impact do you think we could have some of those different approaches you mentioned that we might look to pursue, like vaccines and that kind of thing? Do you see this becoming potentially an important, major new part of our toolbox in preventing breast cancer?
Yeah, that's a huge question. So I think, this issue is very multifaceted. There are structural components, there are equity components to this issue, as well as cultural and society components. So I think from a treatment point of view, it opens up a new part of biology for us to understand women's health and women's immune systems in a better way, and potentially translate to prevention, immune-based prevention for women that can mimic what we see in pregnancy and lactation. But I think importantly, and the WHO has always endorsed this, but it hasn't been taken very seriously, is that women need to have the time and the structural components need to be put in place so women can breastfeed, essentially, or to be encouraged to breastfeed, and that should be promoted. So there's lots of other things that need to be put in place to affect what we've found, and not just treatment and prevention-based strategies as well.
Right.
But there's also general immune factors that can also be put in place. But specifically for our research, it's a very big lens that needs to be looked at.
Right, thank you. So your research spans from the very beginnings of breast cancer right through to following diagnosis and treatment. which is a place that you've really pioneered over the last decade. In some other cancer types, we see more profound responses, I guess you could say, to immunotherapy, like in melanoma, for example. Do you think we're going to get to the point where as many breast cancer patients experience as much benefit from immunotherapies as we might see in other disease types?
Again, you're good with the questions today. I think, obviously breast cancer is a very different disease to melanoma. The causes are very different, and the reasons why melanoma patients respond to immunotherapy treatments is very different. But certainly, I think enhancing your immune system against breast cancer, we know it now works. We have proof of principle that it works, and people are beginning to understand and try to understand why it works. And I think ultimately it will be a multifaceted approach. We need treatments that target the cancer as well, and we also need surgery to remove the cancer because we can see that actually a very tiny cancer has strong effects on your immune system as a whole. So removing the cancer is also important. Chemotherapy or endocrine therapies that target the actual cancer are important, and also things that activate your immune system are also important. I think for other cancers such as melanoma, they have a similar multifaceted approach. It's just they have more emphasis on certain parts of that treatment approach. But you have to come at it at all angles because, once the cancer develops, it's already worked out how to, outwit your body, you know what I mean? Like it has already escaped your natural guardians, and so you really have to really shift the balance back in a dramatic way to get on top of it.
Great point. And so maybe on that idea of kind of advanced disease and this kind of arms race, I have a question for you, Liz. You talked about this junk pathway and this really exciting new discovery you've made about a genomic change that can be a key to understanding how a tumour becomes resistant to one of our most common therapies. How do you think about the future of that? Are we in a kind of an endless arms race with the tumour, or do you think we'll be able to kind of corner the cancer in a way that it can't slip out from our grasp?
I think
tough question, I know.
That is a really tough question, Alex.
Apologies.
Look, I think it's always going to be very hard to corner a cancer. I think they're extremely adaptable. But I think the knowledge we gain from studying all those adaptation mechanisms, we find ways to hem it in. Not necessarily eradicate it, but hem it in. And then when we find that knowledge in more advanced cancers, we can take that knowledge back to earlier cancers and perhaps have a greater chance of eradication. So I think that's the pipeline we're on to try to implement our findings.
Right, thank you. So, Elgene, you told us a lot about hormonal therapies. And could you talk a little bit about how you see these kind of immunotherapies that we've mostly talked about in the setting of triple-negative breast cancer today, but the potential that they have in the other very common subset of disease, these hormone receptor positive tumours, and the kind of place that they're beginning to occupy there?
We do realise that our traditional understanding of breast cancer continues to evolve. So whilst we've previously classified breast cancer as triple negative, HER2 positive, or hormone receptor positive, even within one subset, there are some overlaps with others and that we can learn from other subtypes of breast cancer as well. So one of the studies which Shireen led looked at the use of immunotherapy in hormone receptor positive breast cancer, trying to understand if this strategic approach could apply to a different subtype of breast cancer. And her study, if I may sort of summarize, showed that in patients with this immune inflamed breast environment, even if it was hormone receptor positive, immunotherapy also worked. This is not yet PBS reimbursed for this indication, so we still can't use that treatment. But certainly these sort of trials really allude to really help us understand and break free from our traditional understanding of what breast cancer is and is not. In the context of hormone receptor positive breast cancer, we do know that inhibiting the hormone receptor signaling pathway still remains very important, and there are many new drugs being developed in this setting. And what we've also learned is, like Batman and Robin, a number of you might have heard me use this analogy, that we can actually bring Robin in and to partner with Batman and maybe more efficient in actually treating hormone receptor positive breast cancer. There are some movies where you only see Batman, and for some patients, all you need is anti-hormonal therapy. But there are some patients, such as patients with a medium and high-risk breast cancer, early-stage breast cancer, where if you bring in Robin, which is the CDK4/6 inhibitor, it does a better job than Batman alone.
Thank you, Elgene. Well, we've reached the end of our time today. I think it's been a really wonderful session. I think you'd agree with me. Please join me in thanking our panelists. And Elgene will kindly give out the gifts. Also, the Estee Lauder companies, thank you so much. And finally, congratulate again Dr. Kim on her award. I hope you all have a wonderful afternoon and weekend. Thank you for coming.